We're continuing with the science of bioreactors in this lesson, and we'll discuss the next 3 bullets in this video. We covered HRT in the last lesson, so now let's move on to how temperature impacts nitrate removal. I'm showing a couple years of water temperature data that we measured in the inflow of 6 bioreactors in Illinois. You see drainage water gets down to about 2 degrees Celsius in early spring and peaks out at about 19 degrees Celsius in late summer. We often use an average of about 10 degrees Celsius or about 50 degrees Fahrenheit to approximate the annual average groundwater temperature. And that's a pretty good rule of thumb, but you can see that the actual range of water temperatures going into our tile drainage bioreactors is pretty big from the point of view of a bacteria. Water temperature has been identified time and time again as one of the most important and one of the most clear factors influencing nitrate removal. This early meta-analysis of bioreactor studies showed a break in nitrate removal occurred when you got above about 43 degrees Fahrenheit or about 6 degrees Celsius. To illustrate this temperature concept with a little bit more raw data, this was from a bioreactor study for treating aquaculture wastewater, and their actual aim was to remove formaldehyde from the water, but they also happened to study nitrate. As the time in the bioreactor increased, you see the nitrate concentrations decrease, as we would expect to see from an HRT impact. But those concentrations decrease more for the 23 degrees water versus the 15 degrees water versus the 7 degrees water. I also wanted to show this because aquaculture is a really cool application for bioreactors because their nitrate concentrations tend to be really pretty high. Zooming back out to a recent global review, you see nitrate removal efficiency and percentage increases with temperature to a point, as does the nitrate removal rate. At most of the temperatures I work with, nitrate removal increases pretty generally with temperature, but it is interesting to consider that there is likely a high-end temperature where denitrification becomes less efficient. These high-end temperatures are pretty warm compared to my groundwater. One other temperature-related concept I'll introduce is the idea of the Q10 temperature coefficient. This is a measure of the degree to which a biological process depends on temperature. Mathematically, Q10 is the ratio of reaction rates that happen 10 degrees apart. For example, take the ratio of the nitrate removal rate that you observe at 15 degrees Celsius divided by the removal rate that occurs at 5 degrees Celsius. In this example, that might be around 2, which means that the removal rate doubled at that temperature that was 10 degrees higher. I didn't do an exhaustive review of Q10s reported in bioreactor studies, but these do tend to be on the order of 2 to 3, which means that as water temperature increases by 10 degrees, our nitrate removal rates in bioreactors double or even get close to tripling. I'll make one last point about water temperature by using data from one of the bioreactors we monitored in Illinois. In the top flow rate graph, you see the bioreactor is treating most of the water from the field during this spring period with only a little bit that bypassed with that little dashed yellow-orange line. That matches up with the bottom cumulative flow graph because the cumulative flow is increasing the most during those same months. The bottom line is steepest in about February to May. But what I really want to show you is how the cumulative flow line relates to water temperature. During this time, where the cumulative flow graph is steepest, basically means that where we're trying to treat a lot of water in the bioreactor, unfortunately, our water was the coolest. We can calculate the average daily water temperature for this period by just taking an average of all the daily temperatures, and we know that's about 12 degrees, but that kind of hides the fact or masks the fact that most of our flow was treated at a temperature of less than about 7.5 degrees Celsius. Compounding this issue of cool water is the issue of dissolved oxygen solubility. You know from the first video in this series, solubility of DO increases with decreasing water temperature. So not only are we dealing with cool water temperatures, which slows our bacteria down, this is also the period where we have the most water leaving the field and the most water that we want to treat in our bioreactor. And to make it a triple whammy, cool water also just naturally holds more dissolved oxygen that must be removed before denitrification can start. Moving from temperature to available carbon, in the Bioreactor Basics module, we discussed that most wood chips have about 46 to 48% total carbon. One of the most apparent ways to observe the importance of carbon is by looking at bioreactor performance during its first year versus following years. That first year always has the best nitrate removal. Even early on in bioreactor research, we realized that all of that easily labile carbon, or carbon that was washing off of the wood chips easily, was fueling higher nitrate removal in that first year after a bioreactor was built. One way to think about the carbon required for denitrification is by using a required carbon to nitrogen ratio. This comes a little bit more from the wastewater treatment side, but the carbon to nitrogen ratio required for complete nitrate reduction, that is denitrification, depends on the type of carbon, like how complex or simple the carbon chains are, and how available that carbon is, as well as the CN ratio depends on what type and species of bacteria are present. One handy rule of thumb has been developed where the CN ratio required for denitrification for most readily available carbon sources is about 3:1 up to 6:1, and that's expressed as COD, or chemical oxygen demand, per nitrate nitrogen. We don't often measure COD, chemical oxygen demand, or even DOC, dissolved organic carbon, in drainage water, but it's good to know that these very related ideas exist in the world of denitrification for wastewater treatment. Under cool water temperatures, denitrification is especially carbon limited. So one option that's being explored, especially for bioreactors in cool climates, is dosing with liquid carbon. I think this is really interesting, but the practicalities of running a pump, even a passive pump, in a remote area that's often without electricity, where drainage waters flow rates can vary a lot, the practicalities of all that might be a little challenging. I do see applications for this with bioreactors in cold climates, and I'm especially excited about this idea of liquid carbon dosing for amping up nitrate removal at bioreactors, providing tertiary treatment at small wastewater treatment plants. These small wastewater treatment plants often cannot afford multi-million-dollar denitrification reactors. These wastewater treatment plants are regulated as point sources, so they need to meet legally enforceable discharge permit criteria for nitrogen. If you have to provide the regulatory guarantee that you will meet your nitrogen criteria every month, even in winter months when we know that bioreactors don't work as well due to the temperature effect, carbon dosing of a bioreactor could make a lot of sense. And the wastewater treatment plant operators are used to pumps and real-time management, so this is an idea that holds a lot of promise. Another idea to boost nitrate removal that's related to carbon is the idea of drying the wood chips. Remember, I have said that decomposition of wood chips is an aerobic process. Well, when the wood chips are being broken down, basically what's happening is that their carbon is being released. There's been some nice work that shows when a bioreactor is flooded, then dried, then re-flooded, those wood chips release extra carbon. You see that following as many as 39 drying cycles, this treatment in yellow released extra dissolved organic carbon, DOC, into the water, and that DOC was fueling extra nitrate removal compared to bioreactors that were staying fully wet. The final carbon-related piece I'll mention is that the denitrification reaction produces alkalinity in the form of bicarbonate. This is really more important again from a wastewater treatment perspective, but it's a useful tidbit for your back pocket. In wastewater treatment, that alkalinity is generally really useful because other treatment processes consume alkalinity. And just a refresher, alkalinity is measured as calcium carbonate and helps to buffer the water to maintain a more stable pH. Moving on to microbiology, which I suppose I saved for last because I am not a microbiologist. But of course, the microbes are the workhorses of this process, so it's important to discuss them. The first thing I want to mention is that microbiologically, we are talking about heterotrophic denitrification. This is where organic carbon from our wood chips is consumed as food. All animals and most bacteria are heterotrophs. And you know that putting this in chemistry terms for denitrification, this is where the organic carbon is oxidized as the electron donor. The organic carbon is the food, and it's oxidized to provide energy. However, there's another kind of denitrification, autotrophic denitrification. Autotrophs make their own food. That is, they use inorganic carbon, like carbon dioxide or bicarbonate, to produce their own food. The best example of an autotroph is a plant. They use CO2 to make their own food via photosynthesis. In autotrophic denitrification, inorganic compounds such as sulfur, manganese, or iron can serve as the electron donor rather than organic carbon. In sulfur-based autotrophic denitrification, for example, the elemental sulfur is the electron donor. It gets oxidized to sulfate, and you see the nitrate is reduced to dinitrogen. You also see the inorganic carbon in there as the food source. But autotrophic denitrification is really more of a niche process, so when most people talk about denitrification, they almost exclusively mean heterotrophic denitrification. Lots of work is happening now to better understand the microbiological community in bioreactors. For example, the study showed that the diversity and composition of the microbial community depended on the carbon source. Pseudomonas is always a popular denitrifier, as well as types of Thiobacillus and Micrococcus. But again, we're just starting to scratch the surface of bioreactor microbiology, and for example, this study found that key organisms for denitrification belong mostly to the order Burkholderia. These newer studies are improving understanding of the genes involved in allowing denitrification to happen. The genes NIRK and NIRS, which catalyze the reduction of nitrite to nitric oxide, are used as marker genes to study denitrifiers. Note the temperature sensitivity mentioned here, along with these genes being studied. I think it's safe to say that these communities and the production of these denitrification-related genes fluctuates with seasons and temperature and flow conditions and DOC availability. Remember my attempt to draw these arrows between all of our boxes here? Well, not surprisingly, a lot of them point to the microbiology box. I want to come back to these examples I showed in the earlier video because I wanted to note that these illustrations were very conceptual. There is not a hard break between zones like what I'm showing because not only is the flow constantly changing in our bioreactors, but wood chips are a very heterogeneous and porous media. For example, they have little microsites where bacteria can gather, and those microsites could be anoxic even if the bulk solution is not anoxic. The wood chip surface and internal area is really like a little jungle ecosystem. I show these fungal hyphae, those little spaghetti strings, because I wanted to mention also that fungal denitrification is a thing, but it's very unstudied compared to bacterial denitrification. We know that fungi play a role in bioreactors, but again, this is pretty unstudied. While some fungi can denitrify, I think they're probably more related to breaking down the wood chip carbon into smaller carbon chains and making it more available for bacterial denitrification. But this knowledge about fungus and bacteria in our bioreactors is really just evolving. The last thing I'll mention about bioreactor microbiology is that the idea of bioaugmentation is gaining some ground. This is where really good denitrifiers are grown in the lab and then added to the bioreactor, which really isn't all that done from what's done at wastewater treatment plants for their denitrification reactors. At this point in time, the idea of bioaugmentation leaves me with questions about if these little guys will just get flushed out, if they could really outcompete the native microbial community that's already there. But anything to make our bioreactors work better is interesting to me. Thanks for joining. I'll see you next time.